All right. Thank you very much for having me here. And I hope the people who have seen this talk or a very similar iteration of the European Bioimaging will bear with me. So I'm just going to give you some flavor of what we're trying to do down at Octopus in terms of cryocorrelative workflows. As in, we're trying to buttress the structural biology that we can get from cryo-EM using super resolution techniques in cryogenic conditions. So as you can imagine, my background is fluorescence. I come from the central laser facility, so we work with lasers a lot. But why did we decide to go into cryoimaging? So a few years ago, we were studying the structural biology of a specific receptor, and one of the reviewers basically contested all of our findings because we didn't do it in cryo. And then at that point, we were like, okay, we're going to do it in cryo. But apart from that particular pet peeve, we decided, we looked at the data and saw that like preliminary experiments in cryoimaging had figured out the photon budget you can access. So the number of photons you get for each fluorescent molecule is orders of magnitude bigger than you get at room temperature. Furthermore, you can get blinking from GFP. GFP is the garden variety fluorophore. Everybody has a GFP strain of their favorite protein somewhere in their fridges. So that's it becomes a very accessible technique, or so we thought. You avoid fixation artifacts, of which an example down there using everyone's favorite liquid organelle, stress granules. So you avoid like changing the dynamics of systems and creating aggregation of things that shouldn't aggregate, et cetera, which is what our reviewer 2 pointed to us in our case. And you become instantly compatible with downstream ultrastructural imaging such as cryo-electron tomography. Here is your obligatory cryo-electron tomogram of some vesicles with EGFR embedded in them. So what we, as fluorescence microscopists, faced a whole bunch of challenges. One of the main of which is the cryophotophysics are very poorly understood. There's lots of work in like fundamental physics at cryogenic temperatures, but nobody really cares about what your favorite commercial fluorophore does at cryogenic temperatures. So it's very hard to figure out which dyes will behave the best. So there's a lot of optimization embedded into the whole process. You have to deal with poorer objectives. So normally you would have like really big, very short working distances, oil immersion objectives, which give you diffraction limited resolution of about 200 nanometers. You can't use any of that. So you have to deal with, you have to have a trade-off. And then you have devitrification. If you put too much energy in your sample, the sample starts to change, which is also true at room temperature, but the level of energy you need to put is like orders of magnitude different. And then commercial kit wasn't really meant to do long-term acquisition of inflorescence and was not meant to be stable. So what we did is basically we took the hard way and just developed our own solution. So what we came up with is a thing called a supercell storm technique. So it uses these nano lenses that you can see at the bottom there. It's basically the lens is both the last element in your optical train, so like the last lens in your microscope system, and it's also the cover slip. So you grow the sample directly onto the lens. In 2019, we did the first iteration of this workflow, and we resolved we had a localization precision of about 12 nanometers, which is quite good if you compare with room temperature storm. Is in the ballpark figure of room temperature storm. This was mostly limited by the fact that we had horrible drift, like disgusting drift of microns every hour. So basically this limited our acquisition time, limited our acquisition integration, but it was the best that could be done at the time. In the meantime, we have improved our experimental kit. We've improved our lasers. We've improved our stage. Now we have a stage that has the same drift rating as an SEM stage. So we've managed to do so this is some preliminary data that some of our users allowed to release. So this is Schizosaccharomyces pombe, which is affecting endocytosis. And each of the structures that has been highlighted contains ring structures of actin, where endocytosis is happening. And we calculated the distance between the two sides of the ring, and we think we see structures which are 30 nanometers in diameter. I say we think we see because in the meantime between this and now, we identified further issues with the photophysics that probably undercook our resolution. So we're working our way through understanding the cryophotophysics to actually reconstruct these images with even more resolution. The problem, because we're talking about which problems we're encountering, the problem is that we need the help of like fundamental chemistry and fundamental physics to understand how these things work from first principle and circumvent the problem of, oh, I didn't think about that. Yeah, so this doesn't give you just pretty pictures, because what we want to do is we want to pin down the results that we get from the structural biology but with functional data. So we're interested in co-localizing proteins which are in the same complex. We're interested in figuring out like rare events and pinpointing where we should be cutting. So like where we should be cutting is coming soon. At the moment, we're still doing the fluorescence bit quite a lot. So this is also data from collaborators. They're studying antimicrobial resistance, and they are particularly interested in the vEM complex, which is a complex found in bacteria that inserts bacterial proteins into the outer membrane. So the furthest bit of the bacteria, which is more in contact with the extracellular environment. So this vEM complex is indispensable for bacteria. If they don't have it, they die. And as you can imagine, this makes it a very attractive antibiotic target, because if you can knock that out, bacteria dies, croaks it, there's no way it can recover. But this complex is still quite a bit unknown. It's a very large complex, and it's still unknown whether this complex works in isolation or works with as like a multimeric assembly, works with other proteins all in the environment. So what we did is we've done two-color storm using our supercell method, and we calculated the probability of finding some vEM protein with a client protein and also with a chaperone protein. And based on literature, we are satisfied that we're seeing what we should be seeing, so that the client protein interacts with our target protein for extended periods of time and in specific locations, and that the chaperone protein, which is a periplasmic protein, so it floats about in aqueous medium, has very transient and very rare interactions. So we're not like seeing everything interacting with everything garbage. So what we want to combine this is, of course, cryo-FIB-SEM, obligatory image of cryo-FIB-SEM, which I don't need to introduce because someone else has done for me. This is our main instrument, the Crossbeam 550. So I also don't need to go much in detail in what cryo-FIB-SEM can do in isolation and in combination with cryo-electron tomography. Here is some data that we collected on COVID-infected cells that was then manually segmented by a colleague. Took about a month for this person to do half of a cell. So cryo-FIB-SEM, as has been described by my fellow speakers, is a very powerful technique, which has drawbacks in terms of like how the data is then utilized. There is also like, once you have those volumes, what do you do with the volumes? So in the COVID case, we were interested in seeing what the mitochondria were doing. So we segmented further volumes also by hand to understand in control cells and in infected cells what happened to the spatial distribution of the mitochondria and what happened to their morphological characteristics. So we extracted volumes to extract statistics and figure out what happened. And like, long story short, the mitochondria are damaged, their network is disrupted. In these turnarounds, you can probably see quite clearly that the control cell is a natural network. The infected cell is all broken up. The mitochondria are smaller and all slightly messed up. So cryo-FIB-SEM can give you amazing reconstructions. Then what we do with this reconstruction in terms of quantitative data is also a thing that as a community we need to discuss. Now I'm trying to put it all together so it actually makes sense. So in some cases, for example, when you're studying transient and rare interactions or very specific short-range colocalization of organelles or proteins, how do you target your imaging in cryo-FIB-SEM? How do you decide which regions of interest are the specific ones you want to target? How do you then reconstruct in your volume which organelles are your organelles of interest? Are you going to assume that all of your, say, endosomes are doing the same thing? That maybe you shouldn't, because membrane contact sites, for example, are a specific type of interaction between organelles that is highly regulated in time and space. So at some point, one of the users came to us and said, we're really interested in understanding how endoplasmic reticulum and mitochondria help process the endocytic fate of a specific receptor that binds to Ozempic. So what we did was we made a new homemade super resolution microscope that had an astigmatic lens in the optical train. So an astigmatic lens intentionally deforms the way that each fluorophore looks in your image and encodes the Z resolution in this deformation. So at that point, you can tell from how the fluorophore looks where it is in your imaging plane. So it's above focus, below focus, how much. Because we were trying to understand at which point of the endocytic process these endocytic compartments interacted with endoplasmic reticulum and mitochondria, and in different mutants of this receptor, if this was an enhanced or reduced. So what we did was, we implemented this technique, and we had like an XY localization precision of about 15 nanometers and Z localization precision of about 55. Which is very close to the theoretical limit of what you can do. This was a frame-by-frame resolution. However, we had quite a bit of drift, which limited our capability of doing long exposures. So we're in the process of developing our way out of this problem. But in the meantime, this is the preliminary results of what we managed to do. So we had in the top corner there, that's a aggregate image, aggregate reconstructed image of the whole volume of this insulin secreting cells labeled with Ozempic lookalike with a fluorescent tag and mitochondria. Using this image, we targeted that box there that had the majority of the carriers in positions where we could find them. Took FIB-SEM volume. Here's a still of the FIB-SEM volume. And that's the reconstruction of those specific endocytic tubules that are being endocytosed. So in conjunction with, in yellow, endoplasmic reticulum, and in blue, the plasma membrane. So this is just to give you a flavor of how you would target this specific area to know that these are the carriers you're interested in. Further, the user was really, really interested in later endocytic events. So when the endocytic compartments are being labeled for recycling or lysosome transport. And in this case, we segmented out a whole bunch of examples where we had very close interaction of the endosomes in green with the endoplasmic reticulum in goldish and the mitochondria in pink. And this data can then be analyzed through quantitative methods to assess whether these interactions are enhanced or reduced in disease settings. That's it. That's how far we're coming, and there's more to come in further years, but there is discussions to be had in the community about how you do quantitative analysis and how you integrate different modalities to paint a complete picture of disease mechanisms.
Thanks, Laura, for that Thanks for that beautiful talk. I must admit that manual segmentation is a bit of a tour de force. I'm not sure I would have the emotional resilience to sit there.
Honestly, it was lockdown, so we weren't allowed to come into the lab. But I need to commend the fortitude of my colleague Marta Sinkiewicz, who committed to the project and pulled it to the end.
Any questions?
Very interesting talk. I really like the way you can actually just build a microscope yourself. I was wondering about the first microscope build where you showed you actually have the sample then directly on the last lens in the whole system. So can you reuse this system? Do you then fully need to dissociate the lens from the platinum disc and re-glue it?
No. Long story short, like the glue is basically the same glue they use for satellites, the stuff that goes in space, which is weirdly appropriate, seeing where we are. And the glue is highly resistant to chemical cleaning, so we can take the whole assembly, as long as it's not being fibbed. If it's being fibbed, we need to discard the lens and reuse the disc. If it's not being fibbed, we can clean the lens with highly aggressive chemicals that might explode, and then we can reuse it.
You already answered my second question. How can you fit this without damaging the lens?
You don't. The lens then gets thrown away. Okay.
Any other questions?
I'm just curious in the astigmatis, how deep you get there with the, because this is the single molecule localization strategy you choose.
Yeah.
How deep you can enter in the sample with one shot?
I think it depends on the lens. So it depends on the lens length and strength. I think this one was about a couple of microns, but we chose the plane to be in the apical surface of the cells. So we focus on the apex, and we had.
Do you remember what the objective you were using here? And the quality. I mean, it's a dry objective. It was one numerical aperture.
I think it's a custom one that we had. We basically had made by the manufacturer is like 100 times 0.9. It's the very short working distance objective. So basically, yeah, you go really, really close to the grid, but it's the best that money can buy, basically.
It's still dry objective.
Yes, it's a dry objective. So basically, you're working with 0.9 NA.
Yeah.
In the supercell, the lens itself is the last objective, and it has a numeric aperture of 2.17.
Just get surprised, it's 2 microns. I was thinking about more 700, 800 nanometers. Especially with that NA.
Yeah, I mean,
I just thinking how much you penetrate to be able to match with a focus ion beam.
Yeah, we knew it was just a bit at the top. So we basically used the apical surface of the cell as like a landmark, and then we had like our volume of the fluorescence up there. There are other strategies that we tested, including a thing called a double helix, but then you have like really large PSFs, which limit the labeling.
Maybe you need to talk to William Morner, because they're working a lot. I mean, you know, the Nobel Prize.
Yeah,
and they can go 6 to 8 microns inside by using sort of the double helix, but they have more.
Yeah, there's tetrapods. There's a whole bunch of different things. There's also like some newer stuff that has been presented last year at one of the super resolution, which like uses smaller PSFs and theoretically goes deeper. Yeah. At the time, we had our hands on the astigmatism and the double helix, and we decided the trade-off wasn't good enough for the double helix. We had densely labeled sample.